Good morning, everyone. I'm Vicki Colvin, Brown's Provost, and I'm really happy to be here this morning and welcome you all to this exciting panel and overall our celebration of Brown's 250th anniversary. I'm new to Brown, but one of the reasons I came was for a lot of the exciting interdisciplinary research that's really at the forefront of both knowledge, and that knowledge will help solve some of the world's biggest problems. And I think in our focus on neuroscience through our Brain Institute, we really have a collection of researchers that spans psychologists all the way to neurologists, looking at some of the most fundamental issues about how our brain works. So as you sit here listening to this talk and thinking about that, realize that your own brain is going through a remarkable variety of different actions ranging from the molecular level all the way up to how you form memories and your perceptions and feelings today. So it's one of the coolest things about thinking about the brain is taking a moment to, I think, reflect back on your own brain and what it's doing. So, as you may know, a lot of our interdisciplinary themes that we're talking about today in the different forums as well as across campus broadly are part of our new plan, Building on Distinction, in which we take areas that Brown is already famous for and begin to shape them more intentionally and with even more purpose towards really, both fundamental understandings, so the fundamental science of how our neurons work and how we make memories in this case, all the way into translation, which is how can we actually intervene and treat disease when it occurs in our brain. And it's actually one of the hardest areas right now, I think, in medical science, but it's also one of the most exciting because of the latest sort of mix of both science and engineering that's being applied, and you're going to hear a lot about that today. So I want to introduce our panelists. Our panelists today include Ricardo Dolmetsch, I'm going to wave, who is Global Head of Neurosciences for the Novartis Institutes for BioMedical Research. Ricardo previously served as Associate Professor of Neurobiology at Stanford University in the School of Medicine and Senior Director of Molecular Networks at the Allen Institute for Brain Research, where his work explored the molecular roots of autism and other neurodevelopmental disorders. Galen Henderson is the Director of Neurocritical Care and in the Neuroscience Intensive Care Unit at Brigham and Women's Hospital in Boston and an Assistant Professor at Harvard Medical School. He also serves as a member of the Academy at Harvard Medical School, which is responsible for innovation, mentoring, curriculum design, and assessment of undergraduate medical education. Finally, Melanie Leitner joins us and brings her expertise in supporting and advancing medical solutions for neurological disorders. She's currently working within the ALS Innovation Hub at Biogen and has served as a Chief Scientific Officer at the ALS nonprofit Prize for Life and the first Program Director for Faster Cures, the Center for Accelerating Medical Solutions. It's a really rich array here of both fundamental scientists as well as translational scientists, so I'm sure it's going to be very exciting. The conversation will be led by Diane Lipscombe, Professor of Neuroscience and Interim Director of the Brown Institute for Brain Science. Professor Lipscombe's research focuses on calcium ion channels in different regions of the nervous system and the role they play in chronic pain and psychiatric disorders. I hope you'll enjoy the panel, and I'll now turn the program over to Professor Lipscombe.
Well, thank you very much, Provost. So I just want to make everyone can hear me okay, good. I'm really, really delighted to be able to welcome you all here on this very beautiful fall day in Providence. This is a really exciting program that we have lined up for you, in no small part because of the 3 Brown alum who are sitting here, who are, really our national experts in their own disciplines. So this is tremendously exciting. I'm going to give you an overview of the program so that you know what we'll be doing in the next 50 minutes. So I will do a very quick introduction to the topic. The bios, you have a very detailed set of bios for the 3 participants. Then what I'll do is to pose 3 or 4 questions to our panel here of experts. And then around 10:30, 10:35, if I don't do this, someone at the back is going to jump up and start to yell. And then I'm going to turn over the opportunity to you all in the audience to be able to ask questions of the panel. And then we'll do our wrap-ups for the last couple of minutes. and then you'll be running off to another class with 10-minute gap to be able to get to the next event, which I know that you're all used to doing. Okay. So to just kick this off, I'll say that neuroscience is so exciting because it's a discipline that really sits at the interface of multiple other disciplines. And so the challenge for neuroscience is always at the bench or at the clinic and in development to be able to understand how to work at those interfaces. That's what makes it exciting, and that's what the reason for having these three panelists here, to really talk about those interface, the interface between bench science and clinical applications. How do you get funding to the bench scientists in order to further our understanding of the brain and human health. Okay. So again, just very quickly, you had an introduction to Dr. Melanie Leitner, who is actually an undergraduate in neuroscience in the class of '93, just as I got my first appointment in neuroscience here at Brown. Melanie is here because she's an expert in thinking about how to drive funding to provide for research that is really targeted toward developing cures for really tragic disorders such as ALS, autoimmune disorders, and disorders of the blood. She is, as I won't go into any more details because Provost Colvin already told you her position at Biogen Idec. Dr. Ricardo Dolmetsch is at the point of interface to try and understand how to take those basic discoveries that occur at the bench and then translate those into cures and therapies in his role at Novartis. He was class of '90, also in neuroscience, right?
Yep.
Yeah. And then Dr. Galen Henderson is right at the point of delivery of care in the clinic, in the Neurocritical unit. And so he has the challenge of asking, is the technology good enough, and what do we need? And also interfacing with thinking about how do we train our next generation of physicians to really further the delivery of healthcare right at that point of patient delivery. Okay. So there are their expertise, and I'm going to ask them then to, as I kick off the program, to start to think about these questions. And I'm going to start with Dr. Henderson, Galen Henderson. Oh, I omitted to say, of course, you're the class, MD class of 93? Yeah, okay, at Brown. So let me kick off with the first question. I'm going to direct it to Galen.
Okay.
So in terms of recent discoveries in brain research, what do you think are the most likely to be able to be translated into treatments in the clinical setting?
One of the more important areas right now is, I think sometimes all too often we think of drugs in regards to therapy, but we also should be thinking of diagnostic things with therapy as well. So with imaging is incredibly important, and now we are looking at radiographic markers in regards to traumatic brain injury, concussions, and we are now tracking certain fibers that makes a difference, and hopefully we can develop targets to how some of these fibers are damaged and how they possibly could be repaired. Mm-hmm. So I think imaging is something that has really changed the way we look at the world in regards to neuroscience and even more prognosis of patients. For example, there are certain patients who have severe traumatic brain injuries, cerebral hemorrhage, ischemic stroke, those patients who become comatose for various reasons. At one point, we thought that if a patient was comatose, that was really it. And after a certain period of time, that their recovery was incredibly limited. With modern radiographic imaging now, we do realize that some of these patients are able to do much more than we expected. So I think imaging is one of those particular areas that has really uncovered many potential targets for drug therapies of the future.
Yeah, so that, that's really important, and also, and thinking about education, biomedical engineering.
Correct, exactly.
Is a really important discipline that's really contributing to development in this area. Yeah. Ricardo.
So I would say that for us, the big revolution in neuroscience has been the revolution in genetics. The fact that for a long time, the drug industry tried to generate drugs for these big disorders, depression and schizophrenia. And the thing is that none of these are single disorders. They are a whole bunch of different disorders that have different causes, and they have a strong genetic component. And you know, in the year 2000, it cost us $3 billion to sequence the human genome. And now I can sequence somebody's genome for $1,000. And I don't know of anything else that has decreased in cost a millionfold. And this has now led to the identification of a whole slew of interesting genetic targets for both rare disorders as well as for some more common ones. And the thing that's exciting about this is that, you know, we sort of understand DNA. I mean, we understand what a mutation does. We understand how it changes proteins. We understand that that might change cells. There are, of course, a lot of things that need to be filled in between. But it does allow us to connect something that seems sort of very airy and ethereal, like, you know, moods and ideas, with something very concrete that we can actually do something about. So I would say that if I had to point to one thing that was revolutionary, it is that. But of course, there are a few other revolutionary things, some of which are actually Brown innovations. And one of those, of course, are brain-machine interfaces. Now, this idea that we will be able to introduce essentially, well, we will be able to use computers to reprogram the function of specific circuits in the brain. And there are some just stunning examples of this, how this can be done to, you know, restore function of you know, injured spinal cords, but also for other things like depression and addiction. There is a huge opportunity there. So.
Okay, thank you. Melanie Leitner.
So I was worried I'd be last, so I have to say I agree with both of those very strongly. But just in case I was going to be last to answer this question, one of the things that I think has been very exciting in the neurodegeneration space, which has been a space that unfortunately has been somewhat intractable even to our genetic inquiries, and imaging has made great strides. But I think one of the things that is a new appreciation for looking at these diseases is the idea of propagation, and the idea that these diseases actually can start in a certain part of the brain and then spread throughout the brain. And the reason that this is such an exciting new way, if you will, to sort of think about these disorders is because it suggests that even if we can't stop the beginning of the disease, which often happens 20, 30 years before any symptoms are apparent. Sure, sorry about that. Hello? Is that okay? Okay. One of the reasons I think that this is so exciting is that even if we don't have the opportunity to stop a disease at its onset, which can happen long, long before any symptoms are apparent, it means that we might be able to stop the disease before it moves to another part. Clearly in ALS, which is a motor neuron disease that starts in one part of the body and you lose function and weakness in that part of the body and then it spreads throughout the body, this is very, very obviously relevant. But I think in other diseases like Parkinson's and Alzheimer's disease, which are more, you know, commonly thought about, I think the understanding of this mechanism of spread and propagation is really a potential area for therapeutic development.
Thank you all. So, you know, what I think is really, again, important to emphasize here in the answers is this idea of the importance of technology development and the importance of the biomedical engineering contribution to neuroscience, as well as the interrogation of the human genome to really have us begin to understand how we can utilize that information, the drop in cost, which certainly has involved massive developments in technology for human genome sequencing. And then this idea, of course, that a disease, debilitating diseases, can initiate in one part of the body and then transfer and cause dramatic phenotype and dysfunction in many other parts of the body. So this importance of considering the whole physiology in thinking about disease treatment and cure. Thank you all. So I'm going to move on to ask Ricardo to kick off the answer to this question, then Melanie and Galen. What are the challenges to translating knowledge of human brain function and trauma into clinical treatments. Well, this, I think, is his job now.
Spend the rest of the meal talking about this question.
That describes my job. If I succeed, it will, we'll see. So there are a lot of challenges. I mean, so, you know, let me just give you a little anecdote here. So I was a professor at Stanford for 15 years, and I just this last year took on this job as the head of neuroscience at Novartis. Novartis had cut their neuroscience, and they'd cut their neuroscience not because they had been dazzlingly successful. They cut their neuroscience because they had 20 years of continuous failure. And so the first thing that happened is I went there and I met the chairman of the board, who explained to me that he had been against starting neuroscience again. And that, yes, but that had been forced upon him. And he asked me, so you know, okay, so what are you going to do that's different? And so I had sort of expected this. I had been told that this was a blunt Swiss guy and that I should respond in a blunt Swiss way, so I did. You know, I think that there are at least 4 areas where things are complicated. I mean, the first has been in trying to address diseases where we have some understanding about the underlying pathophysiology. I mean, for too long, we have been trying to develop drugs in diseases we didn't understand at all, just because the market was big. It turns out that just because you have a lot of people selling multiple sclerosis drugs, it doesn't mean you've got to make another one, because there may not be any more targets. So that's problem number 1. So if you can go into diseases that you understand a little bit better, you understand the mutations a little bit better, you understand what it is that they do, that you have a better chance of succeeding. Of course, you have to be prepared to make a drug that's going to address the needs of fewer people, but on the other hand, it might be a great deal more revolutionary for those people than it would be for a broad population, all of whom have different things. I think the second problem is that our preclinical models were frankly horrible. I mean, you know, they really sucked. And you know, just to give you an idea, you know, at Novartis, and this is not just true at Novartis, I mean, you know, this is throughout the industry, we were using these mouse models where, for example, we were trying to predict whether something was going to be an antidepressant based on how long a mouse swam in a bucket, or how long it struggled if you hung it by its tail. And you know, this was, you know, how shall I put this? It was not translatable at all. And the same is true for some of our models of neurodegeneration. We made these models of neurodegeneration. It turns out the brain didn't degenerate at all, and so it was difficult to make drugs that way. So we needed better sort of translational models. There are some key technical issues that have unfortunately not been sufficiently popular in academia, but that are really important for making drugs. Things like, for example, how to get things across the blood-brain barrier. Right, it turns out that if you just simply take a drug or you inject a drug, most of it does not end up in your brain. So one of the interesting experiments you can do if you are an undergraduate is you can inject a mouse with blue dye, and then you can see, you know, once the mouse dies or you kill the mouse, you can then check to see what the brain looks like. It's completely clear. The dye does not get into the brain. And this is a real problem if you want to make drugs, or if you want to use drugs that are more predictable, like antibodies or bigger proteins. And so you're really forced to take a small chemical and optimize it on all these different axes, and that takes years and is incredibly painful. And then the final impediment is the fact that we actually are not very good at running our clinical trials. And that's mostly because we have to be able to measure things. You know, in neuroscience, we have all these soft endpoints. You're trying to modulate somebody's cognition. You're trying to improve somebody's attention. You're trying to improve somebody's mood. You're trying to reduce the number of voices that they hear. And as you all know, these are things that vary over time. And if your only instrument is having people visit their psychiatrist once a month or once every 2 weeks, that's just not granular enough. That's not good enough. That's too subjective. And so we're killed by placebo effects in all of our trials, right? They're huge effects because everybody wants the drug to work. And at the end, we don't know if it works because we couldn't measure anything. So those are the 4 key things.
Thank you so much. And so Ricardo did a good job of summarizing the challenges, and I know that Melanie has spent her whole career thinking about how to really overcome a lot of these challenges, target funding into the right areas. So how do you you know, and for devastating disorders such as ALS and muscular sclerosis. So how do you go about thinking about
It's so much easier talking about the problems than it is about the solutions.
Right.
So, I mean, there are several things that seem to clearly be really critical. One is obviously greater collaboration, public-private partnerships. So it's been very interesting over the course of my career. I've sort of sat in different places within the therapeutic landscape, academic, government, nonprofit, now the for-profit sector. And each of those really has an expertise, and those expertises are somewhat unique to those spaces. And so I think making sure that there's a common language between and among the various approaches to a challenge, and respecting the fact that there are those different capabilities within each of those components of the landscape is certainly one part, I think, of the solution. One thing that has, I think, become more and more apparent is that there is a place, we call it the pre-competitive space, where it's much easier to collaborate. It's much easier for companies to work together, which historically is not always easy. It's much easier for companies to work with academics potentially in that space. And government can also come into that space as well. So I think trying to expand the scope of that quote-unquote pre-competitive space, where there's infrastructure requirement, where sort of you can raise all the boats at one time if you focus on that infrastructure. So it's not, you know, company X with my drug, my disease indication, my approach, where, you know, my bottom line is on the line, but it's really someplace where, you know, if we solve this problem, it helps everybody. So that's really an important place, I think, to focus. Another thing that I've been thinking a lot about over the course of my career is the incentive structures. And unfortunately, we have really misaligned incentive structures. In academia, you know, your career, your success is predicated a lot on where you publish, if you're the first or last author on a publication. It's not always predicated on, you know, how many amazing projects did you help move forward, but which ones were yours, or which ones were you the driver on. And so all those people, for example, in the middle of a publication, don't always get recognized for that. That's not part of your tenure decision, for example. Service sometimes can be, but in general, there's not, the incentives to share data, the incentives to, for very complex challenges like the brain, which is, you know, one of the most complex things ever created, you need a lot of people coming together to address a challenge, and we don't really have good ways to bring all those people together and give them credit for coming together. And so I think that's also been a big area where change could really happen. The last place I'll just mention, I think big data is really part of the solution. So, and what I mean by big data, actually in biology, often big data, you know, the folks in the tech world laugh at us when we talk about big data because, you know, taking everybody's phone records around the world and, you know, all of these other big data challenges are often much bigger than ours. But you know, things about the genome or about phenotyping or even about clinical trial information, the answers are really, you can't identify patterns unless you have enough data points to pull those subtle patterns out. And so again, that's going to really require a focus on the pre-competitive space as well as cross collaboration amongst multidisciplinary and multi-sector expertises.
Thank you both. And actually, in thinking about the answers from Ricardo and Melanie, you know, first of all, having the key problems expressed, and then Melanie thinking about structural solutions and incentivizing. And then you finally mentioned big data as a potential, sharing of big data as a potential solution to this. Makes me think about, again, Galen's passion for thinking about the education of the next generation of neuroscientists and clinicians. And so when you hear their answers, what does this make you think in terms of what we need and how we need to train our next generation of neuroscientists and physicians?
I would like to add what's already been said.
Sure,
in regards to the whole process, for the provider part of things, these clinical trials, we love these clinical trials, but there have been negative trial after negative trial.
Right.
And even when we look at the translational models, when things are evaluated in mice or in rabbits or other animal models, it works. Multiple sclerosis, sometimes for Alzheimer's, types of stroke. But when we try those same clinical trials in humans, we're not getting it right. And it's negative after negative study. So that has been very disappointing. In regards to the education of our next generation, we definitely need to do a better job, and I think there probably needs to be a paradigm shift in the way we're thinking about these overall problems. In getting all of this data, we have a lot of data, but sometimes we actually don't know what to do with the data. And so we need a different way of thinking about how we are going to attack these problems. I agree, definitely should be multidisciplinary, but we need another big change. I think 50 years ago, 70 years ago, after World War II, things really changed for this country and the world in regards to research. I think we need another big break. Genetics has been a big break, but we need to do something else with how we have been doing things.
Well, thank you all. What I'm going to do is now go to Melanie, back to Melanie, and ask you to think about this question, and I think it really dovetails with where you were heading in answer to the last question. So, how should universities, private industry, foundations, where you've had ample experience in all three of those areas, and government funding, of course, how do these various funding groups who want to further research and the advancement of cures in brain, cures of disorders and diseases and injuries of the brain, how should they collaborate together? Do they? And do they need to do better?
Yeah, well, obviously we all need to do better because we have epidemics of disease, of brain-related disease in this country, in the world, and that's a very sort of stark reality that I think we're faced with. So, I mean, how do they come together? There are issues of language, and sort of standards, I think, is one way that you can make sure that people are really talking about the same thing. So, for example, if Galen is looking at his imaging data at Harvard, and someone else is looking at their imaging data in the UK, and they use different standards for that imaging data, they can't actually compare their results. So it's, we have apples to oranges when what we really need is apples to apples. So I think government can be very effective in, to some extent, in helping to set standards. I think industry can also be effective to some extent in helping to follow and provide input and also set standards. But again, I think, it's a lot of it is a literal translation issue where people are fundamentally looking at the elephant from different places. And so, I'm seeing the leg, and you're seeing the trunk, and you're seeing the tusks, and so we need to find a common language, a common space, a common set of standards where we're really all talking at and approaching a problem the same way. I think that the foundation world has actually been somewhat effective in being a convener of this type of thinking, because the nonprofit sector, typically you have people who they're really focused on finding a cure regardless of where it comes from, and they're also sort of outside of, they don't have the profit incentive to the same extent. They don't always have, government can sometimes, there's a lot of levels of oversight or constraints that government has to operate under, and sometimes the nonprofit sector can be have more degrees of freedom to operate in that space. So I think in some very effective arenas like cystic fibrosis, spinal muscular atrophy, the nonprofit sector has been very effective in terms of bringing the players to the table and coming up with a framework for moving forward. Often, again, focused on that pre-competitive space or creating that infrastructure. So I think that there's a really important role for that space to play.
Thank you, Melanie. Ricardo, you saw me looking at you, and I'm going to head to you. But we both live in the you're in the development world now, but in the basic science world of trying to understand how things work at the molecular cellular level. But now you have to think about translating that. So what's the role of universities? What's the role of the
how do you translate things? So this is the thing. We're gaining a lot of information, but the signal to noise in that information is not very good. And so what does that really mean? So by far the largest number of researchers, and I would say the vast majority of the breakthroughs are coming from academia. They're coming from places like Brown. This is where all the ideas come from. This is where all the discoveries come from. And it's an amazing engine of innovation. But it's an engine of innovation that can be tweaked. And one of the ways in which it can be tweaked is by trying to make sure that the information that we produce, that the data is as robust as it can possibly be. And one of the problems is this, that the incentive scheme is not quite right. So in academia, we really value innovation over reproducibility, and that makes perfect sense. That's how you move your career forward, that's how your students get jobs, that's how your postdocs get jobs, that's how people move forward. You discover something new that was not previously known, and that's an important part of what you do. But we have forgotten a little bit that we also have to make sure that whatever it is we find is really true. And so one of the sort of more surprising things that I found when I moved to industry is that we essentially troll the literature for interesting results, and then we try them and we can't reproduce them. So I think that, and so I thought a lot about why that is, and I think, I've been on both sides of this. And I think that one of the reasons actually is because our funding scheme in basic research is a bit weird. We're funding people based on, and we're promoting people based on their capacity to get their work into these high-profile journals, and we don't provide enough incentive for people to repeat things and be thorough, or sometimes we just don't provide enough money.
Yeah, so there's no journal of negative results.
Yeah, no, absolutely. So, I mean, not just negative results. I mean, there's negative results. There is just the fact that, people just don't have enough money to do things enough times. I mean, when I do experiments I don't know, I work, for example, on these mini brains that are derived from patients. We now have this capacity to make neurons from patients. And we do this hundreds of times because we know that we need to do that to make it reproducible. But when I was running a lab at Stanford, it was impossible to do that many times because I would have had to, I don't know, do something to my grad student. So I think that's a place where we can really work. I actually think that we just need slightly better ways of, in fact, in industry, we really need that expertise, right? That deep knowledge that somebody who has focused their whole life on, calcium channels provides is essential because we have a bunch of generalists, okay? They're really good at, making molecules and things, but they're generalists. So you need that deep expertise. But it's actually very difficult to work together with academics because the incentives are not very well aligned. And if we can find ways of aligning them, that would be huge.
Galen, so you at the beginning mentioned the importance of the development of imaging technologies, and that not every treatment involves a drug, and the importance of the biomedical engineering developments there. And so in terms of thinking about how those transitions are made from the basic research into your hands in the critical care unit, what needs to be done in order to facilitate that?
I think one of the limiting things right now is, I agree to what's been said, but also there are still silos out there. And when I think about what happens here at Brown and how interdisciplinary things are. That's something that's really key because we are kind of promoted based on this little splinter area, and that's not necessarily the best thing for society. We could do a better job if we had more of that. And so with imaging, this brings in biomedical engineering. We work closely with physicists in these particular areas, people who have backgrounds within nuclear physics as well, and just finding ways to reproduce some of these results. As what was said before, with some of the imaging modalities that we use, some of the results that we get on one machine, the same machine, built by the same company, we might get a totally different result. And standardization is going to be key, and who's going to take leadership for all of this? And who really owns, who can grab this bull by the horns and say, okay, together we should move in this particular direction. Things that we struggle with.
Thank you. Well, so I'm going to just take a few seconds here to wrap up a little bit, and then I'm going to ask you, as the audience, to think about questions that you would like to ask the panelists. I think it's really, it's you. You're the experts, and you're going to be the solution. And that's why it's so exciting to have this group of 3 panelists here. I'd say what I want to do is as you're thinking of your questions, and if you do, please go ahead and move up to the microphone. I think that what strikes me, though, and I want to make sure that we really emphasize this, the reason for having this discussion about whether I want to have the problems lie, is that the advances in neuroscience have just been so tremendous and are so exciting that the technology is there now. That now the field is moving into this space where, like, how can we move forward even faster, make this much more efficient, and really get to the diagnoses, the cures, the treatments, that we all need. Yes, I'm going to throw the questions. Ask for the audience to come up. I already see that you're there. Didn't see who got there first. I love it.
Good morning.
Could you say your name
yes,
and then please
I'm
ask the question?
Jim Dodenhoff,
Yes.
class of '83, and my wife is an academic neurologist. In fact, she probably would be real happy if I was asking the question, but I'm going to do it anyway.
Please go ahead.
Fascinating discussion. She does a lot of imaging based on the West Coast. So as a business guy observing, there's a question coming, I promise. I wonder, as I've watched her career and seen organizations, and she focuses on MS, seen organizations like the MS Society, very strong. And I just went to a thing 2 weeks ago. We will find a cure for this disease. Yet where I've really seen the gains in MS are in quality of life. And so I guess the question is, this allocation of dollars, is it going to the right places? That's a question. And then the research issues you raise, particularly clinical trials, so long, 10-year trials, where people, brilliant people, essentially are staking their careers on these things and really have a driving interest in successful outcomes. Is there a gap there? And how can we fill the gap better? Are we spending too many dollars on the cure and not enough dollars on quality of life for people who have these kinds of disorders? So that's my question. Thanks.
Good question.
Go ahead. I think I don't think there are enough dollars spent in research, and when we look at the overall numbers of dollars in research, I think it's either flatline or negative. So I think there does need to be more research. In looking at the target, yes, we're looking for a cure, or we do focus on outcomes. But sometimes when it comes to the basic science areas or the translational areas, we have these surrogate markers for outcomes. But the only thing that really matters at the end is the clinical outcome. Is the patient better? Has the patient been treated more effectively than years ago? Are we making headway with this? And I think we need to focus more on those issues, and I think we have focused on those issues, but there needs to be more money into those areas.
I would say in terms of allocating funding, it's a slightly dangerous thing to sort of decide on a kind of global, from the top, we ought to devote more money to this or to that, because in fact, we are really terrible at predicting the future. So the quality of life gains that we've seen in MS, actually MS has been really a success. I mean, I mean an amazing success. I mean, when I was first in, when I was in graduate school, there was nothing for MS. I mean, there was high dose steroids. And now we have a whole set of treatments, including some oral treatments that are really revolutionary. And in fact, at least one aspect of MS, which is kind of the relapsing remitting part, I think we are well on our way to curing that. There are some aspects of MS that still need to be worked on, but I think that's a huge success. But the interesting thing is that didn't come from some sort of effort to improve the quality of life of people who were being treated with steroids. That came from a bunch of work in immunology. So it's difficult to know what is going to work, and we have a system where a lot of things seem to be wasted because you don't know how they're going to turn out, but this is because we're looking at it now. And if you were to look at it in 50 years, you say, I am so glad they did those experiments in flies, right? It turns out actually that almost all the cancer targets are from the fly eye. Who would have thought, right?
Yeah, so thanks. And I know that Melanie, one of the great successes at the company that you're with now is in MS treatment.
Yeah, no, I mean, the other diseases in neuroscience by far would love to be as successful as MS has been in terms of, just having effective drugs. I mean, we don't really have effective drugs in Parkinson's, in Huntington's, in Alzheimer's, in ALS. You asked about sort of the length of time. Part of that is, again, because we don't really have great models of the disease, although MS, I think, has been much more successul in translating, again, probably because it's much more of an immunological as opposed to a fundamental neuro-related disease. But we've been looking and thinking a lot about cancer. And in cancer, there have been remarkable advances, and a lot of that is because they can take the cancer out and do things to the cancer ex vivo. And we can't do that with the brain and spinal cord for the most part. And so I think that also means that we have to do those long trials because we don't really have a way to model the disease on a shorter time scale, and we also don't have sensitive ways to measure the disease as it changes over time.
Thank you so much.
I do want to say something about just the time spectrum, just because if we think about the types of therapies out there over the last 50 years, so we have treatments now for multiple sclerosis, not a cure, but treatments. For migraine headaches, which used to debilitate millions of people, more effective treatments over the past 50 years. Acute ischemic stroke, so things have developed, much of it over the last 50 years as a result of a lot of research. So we do have some.
I think that's an important perspective.
Glass half full.
50 years ago we didn't have much, but now we have much.
Yeah, we can be intractably negative here, but it's not so bad. I mean,
you've been standing there for a long time on the left-hand side, so please go ahead, introduce yourself and ask your question.
Bonnie Friedman, class of '79, and I'm a layman. I didn't take any medical or bio kind of courses in college. It seemed to me that a few decades ago, all the medical work was focused more on treating heart disease and lung disease, the things that we heard more about. And now there's a lot more knowledge for those of us who care about preventing it. And you mentioned that there's an epidemic of brain disease. I was wondering if in the next decade or so, will we learn more about how to prevent brain disease so that we don't need all these drugs you're talking about?
Great question.
yeah.
Maybe I'll just ask a couple of you to respond so we can get through the other questions.
Just to simplify things a little bit more, in thinking about cardiac disease, heart disease, when we look at the number of diagnoses as far as types of heart disease, we can think of about 15, 17, or 20 different diagnoses. When we think about neurological diseases, we're thinking of about 850, 1,000 different diseases. So that's one organ system, the heart, which is a much more simple organism compared to the brain. And some of these diseases for the brain we may be able to prevent. Some are genetically programmed, possibly, but it's different. You're comparing apples to oranges, but we can learn from what they've done.
I was just going to say, it's also been very difficult to identify environmental contributors to a lot of our neuro-based diseases. It's just been much more difficult than diabetes, heart, some of the places where we've made extensive strides in prevention, because we can look and identify those external factors that we have control over, whereas for a lot of the neuro-related diseases, it's been so much harder, although I think there has been some progress in some of them.
Yeah, I mean, the epidemiology of some of these diseases has been getting better and clearer. I mean, for example, in schizophrenia, we know that there's a strong genetic susceptibility. So, for example, if you have a lot of common variants, if you have what people call a high genetic load, your chances of developing schizophrenia are 50-fold elevated over a control. But it turns out that you also need to be subjected to a certain amount of stress. And so we know that stress actually plays psychosocial stress, whatever that means. I have to say it's very kind of ambiguous, but if you look at populations that have migrated from one place to another, they have elevated levels of psychosis. So in a way, that's helpful because it tells you that if you have a susceptibility, you should be extra careful. There are some people we really should protect from these sort of social, psychiatric stresses, and other people who are going to be more resilient. And if we can protect them at these key points in their lives, things will be better. And we in Alzheimer's, actually, we've just started a prevention trial, the first prevention trial, where we are taking people who are cognitively fine, but we have some idea of when it is that they're going to get the disease because they have a specific mutation, and we're giving them the drug before they get the disease because we pretty much know the drug is safe. So we're moving there. It's just simply that these are long, long diseases that you get when you are, much older, and as a consequence, the epidemiology is really hard.
Thank you. Thank you, everyone. I'm going to move to the right-hand side now.
Hello.
Yep.
My name is Susanna Slepian. I'm actually a senior undergrad here studying neuroscience. I'm really excited to have you all here. One technique I've been dying to try is the induced pluripotent stem cell technique, and I'm just wondering, how representative do you think that the brain in the dish actually is of the real brain?
Oh, great question.
That's a great one. So I was talking before about how in cancer we can take things out and we can experiment externally, and that's much harder in the brain and spinal cord. I'm hopeful that iPSCs will be one potential way to do that. There are still obviously a lot of challenges. By and large, those are either monoculture or they're very simplified systems, and they're also flat. So they're two-dimensional as opposed to a three-dimensional, very heterogeneous, cellularly based system. So, and there's also a high degree of variability. I think the field is making amazing strides in terms of trying to figure out what do we need to have, one iPSC line be the same, or one passage will be the same as the next 10. So again, thinking about standardization, there's been a lot of effort going towards that, but it's not really sexy science, and it's not really the stuff that's going to necessarily get you a science paper. So I think hopefully a lot of that will actually come from industry, where it can be scaled, it can be tested, and but I think that that's an amazing technique and technology.
So I'm going to ask Ricardo to comment, but I want Ricardo for the audience to just define really quickly what induced pluripotent stem cells are, so that and then we'll take Melanie's comments in the right context.
So. One of the issues in neuroscience is that, of course, we don't have access to cells of the brain. So one of the reasons that we have been so much more successful in oncology is because you can take a little piece of somebody's tumor, put it in the lab, and treat it, right, as Melanie said. Historically, we just never had that in neuroscience. As a consequence, we were always forced to work on neurons from experimental animals, and it turns out that 60 million years of evolution, which is what separates us from mice, has made a really big difference. The mouse brain is about the size of my eyeball, right? Sometime in around 2006, actually a lot of people were pondering what to do about this. And you know, there was what was sort of a revolution in stem cell biology. So we used to think that differentiation was kind of a one-directional arrow. It was, you know, you started it from an egg and you became a human, and that went in one direction. This is like spilling water. You spill water, you don't unspill water. It turns out that's not true. You can take a cell that has become a skin cell or a blood cell. It is really a skin cell or a blood cell. And you can put these factors that change the folding of the DNA, and you can convert it into a stem cell. And then you can take that, and these are called induced pluripotent stem cells. And then you can take those stem cells and then subject them to various factors, and you can have them form neurons. And actually now we actually can form little brains with little layers, and we have been doing this for, you know, almost a decade. And now we have whole incubators full of these little brains from people with different kinds of mutations, with different genetic loads. And it's, well, you know, I would say that we have gotten to the point where, especially over the last maybe 6 months, where we know very much what kinds of cells they are and what level of development they are. So I think, look, there are a lot of challenges, but it is far better than anything we had before for some kinds of diseases. It is not the kind of thing you want to use if you want to understand circuitry, but it is perfectly fine if you know, what you have is some mutation that you think causes, you know, some problem in some aspect of a cellular function.
Thank you. The bell is going to toll very quickly, and so I think we can take like one more very quick question from I don't know who was standing there first, and there won't be any more questions on the mic, but I know that the participants, I'm sure, would be willing to stay behind and have a discussion. I don't know if this room is being used. Someone help me? Yes, okay, all right. So we really need to wrap up. Really, maybe pose your question really quickly, and then I'm going to ask the participants to just wrap up in answering the last question.
Sure.
Why don't we each pose our question, and maybe
then they can wrap up. Okay.
Eleanor Bachrach, class of '65. Much of what you've been talking about, well, there are going to be huge costs going forward to some of these diseases, many of which with longer lifespans will come to fruition, so to speak. I'm particularly concerned in my age group about Alzheimer's. Would it make any sense at all to employ some of these diagnostic tools, whether genomes or imaging, earlier on a routine basis? I mean, sort of like a colonoscopy or something. So these things might be staved off.
Okay. Great, thank you. And then is there a relationship to your question to this?
No, my question, Jeffrey Anderson, class of '84, my question wasn't about the diseases, but you know, there's a lot of stuff going on in the news today about, I have an 8-year-old who wants to score all his goals with a header. In soccer, we have the NFL who's kind of in denial about concussions, and we have parents who are trying to get sensors on helmets, and the school boards are removing them and so forth. So I'm curious what, you know, your opinion is of that particular area of brain injury.
Fabulous. So I'm going to ask, you literally have like 10 seconds each. and before they wrap up, I really want to thank you all tremendously for being here. Go ahead, Melanie. Galen's going to be the closer.
Unfortunately, unlike with colonoscopy, we don't really have a way to treat. So expending a lot of dollars on diagnosis might not be the best approach regarding CTE, chronic traumatic encephalopathy, and this idea of concussion-induced brain injury. I think it's something that really needs a lot of investigation. It's still very early. It's still relatively controversial. It's great that the NFL is putting, I think, $60 million into it. It probably could use more than that, but it is definitely, at least based on what I've seen, I believe it to be real. There's some biology behind that we really need to understand and protect our kids from.
Thank you, Melanie. Ricardo, 10 seconds.
Yeah, 10 seconds. Well, you know, I think that with regards to diagnosis, it depends. It's a sort of cost-benefit analysis, right? So there are some diagnostics that are now sufficiently cheap that we should probably do on a lot of people. Like, for example, sequencing the exome is now sufficiently inexpensive that you might want to do this in, you know, people who are of a certain age at certain risks. There are, of course, a lot of other diagnostics that you probably would not want to do in everybody. Now, as to whether you can act on them, I'm not so sure. We can't act on them now, but it's a good idea to have all that information.
Thank you, Galen.
In regards to screening for Alzheimer's and things like that, I think it is something important to know how a person should plan the rest of their lives, because we do have some patients with genetic diseases that at a certain age they tend to get worse, and there's a lot of ethics involved in whether they should get certain tests or not. So it's something to be considered at a certain point. In regards to traumatic brain injury and traumatic encephalopathy, I think that there's nothing that can be done externally with a helmet that's going to make a difference based on what happens physiologically within the cranial vault. And even when we think about the long-term aspects of things, our first case was not really with the NFL. If you think about boxers and dementia pugilistica. So there are a lot of issues. Football has just gotten a lot of press over the past couple of years, but we've seen this in boxers for over 100 years. So we can learn some lessons from those patients.
Thank you so much, and thank you all for being here.